afternoon and welcome to Critical Care Grand Rounds. My name is Asma Zainab and I'm an intensivist at Cardiovascular Surgical ICU at DeBakey Heart and Vascular Center. Today we are presenting our third in the series of Grand Rounds offered by Critical Care Centers of Excellence at Houston Methodist, and the topic of today's discussion is acute lung inflammation and ARDS. And I have the pleasure and honor of hosting Dr. Diego Martin and Dr. Joe Garcia here at the studios. I will start with introducing Dr. Martin, who recently joined Houston Methodist as the chair of Department of Radiology. He's also the medical director of Houston Methodist Research Institute Translational Radiology Imaging Center. He joined from McGill University, where he served as chair and chief of Department of Diagnostic Radiology. And before that, he served as a tenured professor of medical radiology, biomedical engineering, and physiology at University of Arizona School of Medicine, Tucson. Now I will request Dr. Martin to present and introduce Dr. Garcia.
Well, thanks very much for that kind introduction. It gives me a tremendous degree of pleasure. It's an honor to be able to host Dr. Garcia and to have him present Grand Rounds today. Just a few words of background. So Dr. Garcia is the Dr. Merlin Duval Professor of Medicine at the University of Arizona. He's had over 25 years of continuous NIH funding, including multiple large grants such as program project awards, over 600 publications, over 50,000 citations. His Google Scholar H index is 112, and in 2011 he was inducted into the National Academy of Medicine. He has numerous entrepreneurial activities, which include 10 patents, and he's also the CEO of a biologics company. He previously was the Senior Executive VP for Health Sciences at the University of Arizona, interim dean, and previous to that he was at the University of Illinois, where he served as Vice Chancellor for Research and was also the Chair and Professor of Medicine at the University of Chicago. He was the Director of Pulmonary and Critical Care Medicine at Johns Hopkins, and prior to that, Associate and then full Professor of Medicine, Physiology and Biophysics at Indiana University. And really to make sense of all of this, he's now coming home because he was, in his earlier years, Assistant Professor of Medicine at the University of Texas Health Center in Tyler. So in some ways, this is a coming back to initial roots, and for us also it's an opportunity to explore possibilities of synergies and collaborative research. We have a number of projects working with Dr. Garcia in his lab, and there are a number of discussions ongoing that I'm certain his talk will only further promote in terms of potential collaborations with a number of our key scientists here at Houston Methodist. So Dr. Garcia, with no further delay, I could keep going. Your CV is quite lengthy indeed. It's a tremendous honor and privilege to be able to have you here today. Thank you so much.
Well, thank you so much, Dr. Martin. I want to thank also Dr. Masoud for the invitation to be here. As you allude to, yes, I've had academic appointments in the state of Texas, but more importantly, I'm a native-born Texan. I'm from El Paso. I went to College of Medical School in Dallas, and so it is quite rewarding to be able to come here and present and be part of this fantastic institution. So let me just begin by saying it is a pleasure, and what I'll be talking about is very germane to the goals of the Center for Critical Care. Before I do that, let me just have the prerequisite disclosure that, as Diego just mentioned, that I am the CEO and founder of a biologics company, a biotech company called Aqualung Therapeutics. The assets in this company are be part of what I'll be talking about today, and I think they're very exciting to, I think, not only our company, but also obviously the academic community as well. So without knowing exactly the constitution of all the folks that are listening to this presentation, I think it's pretty clear, given that this presentation is part of the Center for Critical Care, that folks in the audience are very much aware that critical care illnesses, and it's kind of depicted on this slide, which doesn't include all sorts of critical illnesses, but it does include sepsis, trauma, acute respiratory failure, etc. They are a very substantial portion of the U.S. healthcare dollar. And that, in fact, was the situation well before the COVID-19 pandemic. So I will have to say the thematic underpinnings for the talk I'm about to share with you is the following. It's that activation of these evolutionarily conserved inflammatory networks leads to multi-organ dysfunction, and it's that multi-organ dysfunction in critically ill patients that drives mortality. And unfortunately, in our ICUs, unlike other disciplines such as oncology and cardiology, where breakthrough drugs have emerged, PD-1 inhibitors, for example, or Herceptin statins, every drug in the ICU is generic. So there's a major unmet need for novel therapies to address the mortality in our ICUs. So the focus of this talk is, in fact, one of those critical care illnesses, ARDS, acute respiratory distress syndrome, well known to members of our medical community. This is a highly inflammatory disorder, primarily due to sepsis and trauma, but also substantially contributed to by gastric acid aspiration, as well as pneumonias. And obviously, in the pandemic landscape that we live in, the numbers of cases of ARDS in the U.S. has gone up substantially from the 500,000 per year in the pre-COVID-19 era. And mortality has also risen. The mortality pre-COVID was 30 to 40% mortality. It's obviously higher, and it's obviously higher in individuals that are elderly and have risk factors for other comorbidities. So the pathophysiology of ARDS is the fact that the vasculature in the lung becomes leaky as a result of the inflammatory process. That leads to alveolar flooding. This leads to reductions in oxygen delivery to vital organs, and hence the multi-organ failure. Lungs that are full of fluid represent a major task for respiration, and so the work of breathing is so substantial that there's a need for mechanical ventilation. Current treatments for ARDS are merely supportive care. Lowered tidal volumes on the ventilator or prone ventilation is helpful. But there is a major significant unmet medical need for FDA-approved drugs. It's very important to remember that in the ICU in patients with ARDS, that while the mechanical ventilator is a life-saving intervention, the mechanical ventilation and the mechanical stress that is derived from being on mechanical ventilation is also a major contributor to ARDS mortality because it's a major driver of inflammation. The studies underpinning that observation emanated back in the years 2000, when this New England Journal article was published by a multi-center trial led by the Acute Respiratory Distress Network. I was a protocol chair for the ARDS network, and this landmark study, it was really driven by folks that were part of my division when I was chief of pulmonary at Johns Hopkins, really led to the awareness that mechanical ventilation contributed to mortality. And by that I mean, if you compared conventional tidal volume ventilation, which is 10 to 12 mils per kilogram, to what was studied in this particular protocol, 6 mils per kilogram, you could see that there was a significant mortality benefit, survival benefit in individuals that were receiving the low tidal volume ventilation. And this translated to less inflammation when you measured biomarkers and cytokines such as interleukin-6. But the real question became, what's the mechanism that underlies this survival benefit in low tidal volume ventilation? Or stated another way, how does mechanical ventilation actually contribute to increased ARDS mortality? Well, if you look at CT scans that are done of ARDS patients, unlike the chest X-ray, which is fairly homogeneous, If you look at the CT scans, you can see that there is great regional heterogeneity in the CT scans. There's areas of relatively unaffected parts of the lung that are overdistended when they receive a disproportional amount of a ventilator-delivered breath. And there's areas of the lung that are flooded and collapsed and do not open during a delivered breath. But importantly, there's these areas of opening and closing, the alveolar units that open with the breath and collapse. And it's this cyclical mechanical stress delivered by the ventilator that also contributes to the inflammatory process. So we wanted to study this in my group, try to understand mechanistically what might be driving the mechanical ventilator-induced inflammatory process. And to do so, we developed multiple models of inflammatory lung injury. As you can see on the slide here, we use mice, rats, dogs, pigs, and human cells exposed to mechanical stress. And we exposed these animals to a combination of lipopolysaccharide to simulate a pneumonia, and also delivered high tidal volume ventilation to simulate the mechanical stress of ventilator-induced lung injury. Following a period of time where these animals were exposed to the ventilator and to LPS, we retrieved RNA from the lung tissues of these animals, and then we hybridized this RNA to chips, microarray chips as they were when we were conducting these studies. RNA-seq is what folks use now, but microarrays were what we did to survey the entire genome for expression of genes that might be contributing to ventilator-induced lung injury. And then through a series of bioinformatic analyses that we were able to identify selected ARDS and ventilator-induced lung injury candidate genes that we could test for their contribution to the overall course of ventilator-induced lung injury. So, to give you an example of this strategy, here's a canine model of ARDS. So in this model, a bronchoscope is instilled into one lung, and that lung is lavaged multiple times to remove surfactant and make it very prone to have increased injury when placed on mechanical ventilation. And the other lung is left unaffected. So you can see from the CT scans in this canine model that the whited out lung is the injured lung, and the non-whited out lung is the uninjured lung. And the red circles represent areas of the lung where we retrieved tissue, isolated the RNA, and then performed genome-wide expression profiling. When we did this, we identified that we observed that control lungs had very little gene expression and the injured lung had very high levels of gene expression. So this is what's called a heat map, and if you're not familiar with this, let me just tell you that heat maps, every row is a different gene and every column is a different animal tissue from a different animal in which we measured gene expression. The color green denotes the fact that gene expression is reduced, and the color red denotes the fact that gene expression is significantly increased. And you can see there's very dramatic differences between control lungs and injured lung, and even in the injured lung you can see there's heterogeneity between the apex and the base. Now, there's a lot of genes that are shown on this heat map, and the gene that we ultimately began to focus on is a gene called NAMPT, or also known as PBEF. And we wanted to make sure that we were having a good idea on which genes to select. And so NAMPT was selected primarily because it was the highest expressed gene of all the genes on these microarrays. So just to give you a sense about the uniqueness of identifying this gene, let me just show you a bioinformatic analysis that we performed. So we took all 70 of these microarrays that we had done, and we blasted genes against a tool called PubMatrix. So after we identified a number of genes that satisfied our very stringent criteria of increasing expression or decreased expression and a significant p-value in our models of ventilator-induced lung injury, these genes were blasted. We had about 50 genes, and we blasted them against the following PubMatrix terms: lung, lung injury, mechanical ventilation, endothelium, and pulmonary endothelium. So when you do that for example, two genes that were very highly significant were interleukin-1 beta and interleukin-6. And you can see if you blast those genes against all of the PubMatrix, all of the papers that are present in PubMed, you find out there's quite a number of manuscripts that are published with these that contain these two genes. But among the 50 genes that we identified through this process were a handful of genes also very significantly dysregulated in these microarrays. But when we blasted those genes against these PubMatrix terms, you can see there were absolutely no papers whatsoever that incorporated the terms lung injury or mechanical ventilation. So we viewed this as being a way to identify novel genes, perhaps novel targets, perhaps novel biomarkers that would be related to ventilator-induced lung injury. So I will tell you that even though there's no papers there at the point that we published this paper in 2004, we have subsequently published a number of papers on each of these genes, subsequently tying these genes and their gene expression and their functionality to ventilator-induced lung injury. But the focus of this talk for the remainder of my time is going to be on this gene, NAMPT. I'll tell you a little bit about what NAMPT does in a second, but you can see here when we did PubMatrix blasting, there was absolutely no papers linking the expression of this gene to any of these PubMatrix terms. So let me tell you what this gene is. It's an enzyme. It's a metabolic enzyme called nicotinamide phosphoribosyl transferase. And the reason we didn't find any publications linking it to lung disease is because there was only 8 papers in all of PubMed that were associated with this gene. You know, now there's more than 400. So it was our report was the first example of this gene being associated with lung disease. What this gene does, what this enzymatic activity does, is it regulates NAD metabolism. And it does so by moving nicotinamide mononucleotide to NAD and functioning in very important NAD-dependent enzymes such as the sirtuins. But the reason that NAMPT is called a cytozyme is because in addition to its enzymatic activity, it also functions as a damage-associated molecular pattern protein, or a DAMP. This DAMP binds toll-like receptor 4. This is a publication where we showed that this protein NAMPT binds to toll-like receptor 4, and in doing so elicits a very robust inflammatory process. We examined where and what cells NAMPT was being elicited from, and these studies identified 3 major cells that contribute to NAMPT secretion. Those are lung epithelial cells, lung endothelial cells, and inflammatory leukocytes. And in the lower left panel, you can see that the marked increase in NAMPT expression, which is shown in green, compared to dogs that are receiving spontaneous breathing. When you place a dog on high tidal volume ventilation, you can see that the NAMPT expression in the endothelium is rather substantial. On the right, we have mice that are also exposed to ventilator-induced lung injury, and the brown staining is in fact NAMPT. It's really reduced in controls and markedly increased in ventilator-induced lung injury, and we published this a number of years ago. So we know a lot about the gene. We've sequenced the gene, and we understand mechanistically how we think it contributes to ARDS and ARDS mortality. And the mechanism involves what I'm going to show you on this particular slide, which is the NAMPT promoter in response to mechanical stress, viral or bacterial infections, hypoxia, a number of injurious stimuli cause the NAMPT promoter to be activated and cause NAMPT to be secreted into the circulation. In the circulation, eNAMPT, or extracellular NAMPT, binds to toll-like receptor 4. This is a main innate immunity receptor, as many of you know. It's a pathogen recognition receptor that is a guardian for responses to invading bacteria and viral organisms. Once eNAMPT ligates toll-like receptor 4, this this launches what's known as the cytokine storm, because expression of multiple genes encoding cytokines and growth factors that now show up in the bloodstream, and in doing so they create this systemic inflammatory response that leads to organ inflammation and organ dysfunction, as shown on the slide here. Patients with ARDS, as many of you know, die of multi-organ failure. They don't die of respiratory failure. The ventilator is able to keep patients alive for quite a long time. But when systemic organs such as the liver and the kidneys and the cardiac system begin to fail, mortality goes up. So our strategy here is to, in fact, use a biologic, a humanized monoclonal antibody called ALT-100 to bind circulating NAMT, and in doing so preventing ligation of the toll-like receptor 4. And by removing and neutralizing eNAMT, we can block or dampen the cytokine storm. So to underscore and perhaps provide substantial supporting evidence for this importance of NAMT, we evaluated peripheral blood mononuclear cells and compared gene expression in ARDS patients that both survived in one group and failed and succumbed to ARDS in another group, at a total of 55 patients. And then we looked to see what genes differentiated survivors from non-survivors. And when we did so, we used those genes and plugged them into pathways that might be differentiating between survival and non-survival in ARDS. And when we did that analysis, what we found is the most significant signaling pathway that was dysregulated between survivors and non-survivors was, in fact, the toll-like receptor signaling pathway. The receptor pathway that NAMT binds is toll-like receptor 4. So the mechanism, this may be a little bit too in the weeds for a number of you, but I think it's very interesting to note that toll-like receptor 4 is a fairly promiscuous receptor. A number of molecules can bind and activate toll-like receptor 4. For example, lipopolysaccharide, which is a bacterial endotoxin product, uses toll-like receptor 4 to activate the pathogen recognition system. But LPS, when it binds to toll-like receptor 4, cannot activate the receptor by itself. It needs a chaperone protein, a cofactor, that's known as MD2. As you can see here, MD2 is what binds the receptor, and LPS binding to MD2 is what activates the toll-like receptor 4 and elicits the inflammatory cascade. Well, NAMT also binds to toll-like receptor 4, but unlike LPS, NAMT can activate the receptor without the need for MD2. And so we pondered, how is that NAMT able to do so? And we found was that computational modeling of the NAMT protein identified two regions, a loop region and a LPS-like region, that were very similar to MD2. So you can see here that NAMT and MD2 share structural similarities computationally. And then when we looked at the amino acid sequence, we found that NAMT and MD2 shared high homology in the amino acid sequence that binds to the toll-like receptor 4. And that's shown computationally in this diagram, where red is the LPS-like binding loop and green is the MD-like binding loop for toll-like receptor 4 binding to NAMT. Now, we know a lot about what drives NAMT expression. NAMT, in response to mechanical stress, as I've shown you, inflammatory mediators like interleukin-1 beta, growth factors like VEGF, all of these signals, including oxidative stress, cause the NAMT expression to be increased via transcription factors that we've identified and have published on. These transcription factors go to the nucleus, where they bind to promoter elements in the nucleus of this gene to drive NAMT expression. NAMT expression is also regulated by epigenetic mechanisms, in that the promoter is heavily demethylated in ARDS, and specific sites of DNA methylation we published out. It's also epigenetic regulated by microRNAs. So we've identified specific sites in the 3' UTR portion of the gene that bind to specific microRNAs as shown on the slide here, and thus regulate NAMT transcriptional activities in this manner. The overall outcome of this driving of NAMT transcription is more translation of NAMT intracellularly and then secretion of NAMT into the extracellular milieu, including into the circulation. Now let me just provide a little proof of concept. Now we know that NAMT expression is linked to ARDS, but is it a major pathologic target for inducing ARDS-like outcomes? So this is as I mentioned to you before, common mechanisms for inducing ARDS in addition to sepsis is trauma. So we, in collaboration with folks in San Antonio at the Fort Sam Houston Institute of Surgical Research, we conducted studies where we exposed rats to a blast in a shock tube that only the Army could have. They have a shock tube that delivers a specific blast to an intact animal, rat or pig, and in doing so, because the lung's full of air, it stimulates major injury and inflammation in this animal. So I'm showing you in A, B, and C. A is a macro look at the rat lung after this exposure. B and C are also histologic analyses. And so what we do is we expose the rat to this blast, wait 30 minutes, and then we put the rat on a ventilator for 4 hours. And you can see there's a tremendous amount of injury here. And the inset in panel C is what a normal rat lung should look like. And you can see that there's substantial edema and inflammatory cells in the lung in this model. Now we expose a different rat to exactly the same blast, also put it on the ventilator, but in this rat we treat them with this polyclonal antibody that neutralizes eNAMT, and we deliver the antibody before it goes on the ventilator. And the difference is, you can see there's a major reduction in inflammation both macroscopically and microscopically, and even though there's still substantial injury, you can see this animal has marked attenuation of the inflammatory response that we can measure into a lung severity score, which shows that the polyclonal antibody has significantly reduced injury in this rat model. So because of the validation and support for using an eNAMT neutralizing approach, we embarked on identifying a potential therapeutic target in people, and we started with developing an antibody, mouse antibodies, that could bind to eNAMT and neutralize it. So we developed these and developed murine monoclonal antibodies. We took our top 2 murine monoclonal antibodies from these mice, 1093 and 1076. We humanized the monoclonal antibodies, screened those, optimized the sequence, and long story short, we identified our lead monoclonal antibody, which is ALT-100. We have subsequently began stable cell line development with a company in the UK called Fusion Antibodies, and now we have gotten to the point where we have our antibody, it's in stable cell lines, and now we are ready to produce this antibody, and we've started manufacturing of this antibody, and the long and short of it is that we're very excited by the fact that we have conducted a number of FDA studies required to get approval for use in humans, and we're going to start phase 0/1 clinical trial in humans, first in human, at the end of this calendar year. So we have a lot of scientific rationale for use of the antibody, and now we're going to have the antibody for use in people. We think by the end of the year we'll be using it in patients with ARDS. So what's the data that supports using this antibody in ARDS? Well, we started with a mouse model, and this is a mouse that's exposed to LPS down the trachea, so it has an LPS-induced pneumonia, and we treat it. It also is placed on a ventilator for 4 hours, and what you can see here is that delivery of the antibody before going on the ventilator attenuates injury as measured by the severity scores on the right panel. We also developed a endothelial cell specific knockout mouse to demonstrate that eNAMT coming from the endothelial cells is really of primary importance in this model. So when you expose an animal to the ventilator, the eNAMT coming from endothelial cells appears to be very, very critical to the induction of that inflammatory response, because eNAMT knockout animals are protected. And we published this paper in the European Respiratory Journal in December 2020. And what I want to bring your attention to is two individuals that were co-authors on this paper. Dr. Diego Martin, who's to my right here, and Dr. Zonglin Liu, who has joined Dr. Martin's Department of Radiology here. These were two very, very important collaborators on this project, and we, with their help, were able to show using a monoclonal antibody technetium technique that NAMT expression was markedly increased in the injured lung. But I think the most compelling data supporting use of this antibody in people comes from this model. This is our porcine septic shock ventilator-induced lung injury model, where we take Yucatan mini pigs into basically a porcine ICU. So these pigs are instrumented, they're hemodynamically monitored, they have ART lines, they have 2 venous catheter lines in place, and we monitor all parameters just like we would in an ICU. And once they are stabilized, instrumented, and at baseline, we then infuse LPS intravenously at time 0, and then the animal is placed on high tidal volume ventilation for 12 hours. And very easily you can see that compared to controls that are not receiving LPS and ventilator-induced lung injury, pigs that are exposed to this challenge have tremendous inflammation. There's some hemorrhage there. You can see there's lots of edema. Then we treat these animals with a monoclonal antibody delivered either at 2 hours after the onset of injury or 6 hours after the onset of injury. And as you can see from the H&E histological analyses here, there is a significant attenuation. These are still injured lungs, but compared to the untreated animals, this is a really substantial attenuation of inflammation. This is corroborated by when we measure the number of neutrophils in bronchoalveolar lavage fluid. You can see 2-hour or 6-hour delivery, the antibody substantially reduces lung inflammation. Lung severity scores are reduced by 2 and 6-hour treatments in a time-dependent fashion. And we think this is very interesting too. So if we measure lactate levels in this pig model, lactate levels begin to be significantly reduced in the 9 to 12 hour period after the onset of LPS and ventilator-induced lung injury. Oh, sorry, I went the wrong way. The other 2 indices of inflammation here that we measure NAMT levels in the plasma. You can see NAMT levels are significantly reduced by antibody treatment, as were levels of interleukin 6. We think these data are very interesting as well, because when we wanted to measure indices of lung edema. So panel A here is showing you the amount of protein in bronchoalveolar lavage, and this is an index of vascular permeability and eventually protein from the vasculature arriving in the alveolar space. And you can see that it's a tremendous increase in animals that receive PBS, and in animals that received our antibody, that there's a very time-dependent but significant reduction in BAL protein. We used another technique that's well known to perhaps a number of you, where we just did ultrasounds in these pigs and then looked for B lines, which are the index of lung edema, and we counted the number of B lines in these pigs. And you can see in panel C that the number of B lines was quite elevated in pigs that are injured with the LPS ventilator-induced lung injury model. In contrast, pigs receiving the monoclonal antibody had a marked reduction in number of B lines, indicating that lung edema, in fact, was reduced in these animals. And that was corroborated as well, but we measured static and dynamic compliance, and indices of lung stiffness. So what you can see that in the time dependent in panels D and E here, at starting at 5 to 8 hours after the delivery of the antibody, the reduction in dynamic and static compliance begins to be improved in animals receiving the antibody, so that it's highly significant by 9 to 12 hours after the onset of injury. And then finally, in panel F, we're interested if we had evidence of renal injury in this model. And so we measured a biomarker for kidney injury, lipocalin-2, and you can see lipocalin levels are increased at 12 hours in untreated animals, and the levels of lipocalin, an index of kidney injury, acute kidney injury biomarker, is reduced by the antibody. And we further evaluated this by doing histological assessment of kidney tissues. And you can see normal renal tissue on the left and LPS, ventilator-induced lung injury, challenged tissues on the right. In the absence of any intervention, what you can see with your red arrows is there's duct dilatation and frank evidence of frank necrosis in the left side of this particular slide. And in animals receiving our monoclonal antibody that neutralizes ENaPT, we saw no such evidence of any necrosis, and duct dilatation was markedly reduced. So all in all, we're using this preclinical model in a porcine model of ARDS. There's multiple parameters here that we think we're reducing inflammation, improving physiologic parameters in these ventilated animals. And we think that by reducing lung edema in patients, this will result in reduced days on the ventilator and improved survival. We evaluated very quickly. We've evaluated signaling pathways. This is taking lung tissue from these pigs and blotting for phosphorylated NF-kappa B, which is a signaling transcription factor of inflammation. And you can see that there's tremendous NF-kappa B activation in injured animals, and in animals receiving our monoclonal antibody, that is markedly reduced. Similarly, AKT is a signaling pathway very much involved in regulating inflammation and vascular permeability. AKT levels are nearly absent in the injury model, and there's a significant AKT nitration, which leads to degradation of the protein. This is obviously also attenuated by our monoclonal antibody. And then AKT works in concert with mTOR, which is a signaling pathway. The mTOR-Richter pathway is also a very important signaling pathway with AKT. And you can see Richter levels are lost in the injured lung and restored by antibody utilization. And then I think another corroborating piece of data in humans is when we looked at the gene expression in differential gene expression in survivors and DNA methylation in these survivors versus non-survivors. So what this shows you is that when we analyze what genes are being differentially demethylated in ARDS, they fell into the following 5 top categories, which were regulation of mTOR, mTOR signaling, PI3 kinase, AKTOR, and mTOR signaling, and mTOR complex 1 signaling. Therefore, the signaling pathways that are epigenetic regulated in ARDS are very amenable to being improved by reductions in ENaPT via a humanized monoclonal antibody. In additional supportive studies, we've removed PBMCs from patients with ARDS and incubate them with this antibody. What we found at baseline, that gene expression of toll-like receptor signaling and NF-kappa B signaling were markedly increased in ARDS patients compared to controls. And then when we incubated those PBMCs, peripheral blood mononuclear cells. With our monoclonal antibody, we identify the marked attenuation of these genes by the antibody, suggesting that the antibody has the capacity for downregulating these inflammatory signaling pathways in humans. So where is this all leading to? Well, of course, we believe that we're on the cusp of perhaps having a novel therapeutic for sepsis and ARDS and COVID-19 induced ARDS. But what's the big challenge here? The big challenge is that ARDS and sepsis represents a landscape of multiple failed trials. And if you think about sepsis studies done in 10 years ago, 15 years ago with Xigris, which was an Eli Lilly compound, activated protein C, that failed. 2 or 3 years ago, a gamma interferon molecule was tried in phase 3 trials in ARDS. It failed. And basically in the COVID-19 pandemic, we've repeated 20 years of ARDS research in about 6 to 12 months. And again, we have yet to see a major therapeutic advance emerge from these clinical trials. And so why is that? Well, ARDS is an extremely heterogeneous disease, and you need many patients to show an effect. That's a big challenge. And many promising drugs were derived primarily from mouse studies, and I think there's very, very well appreciation of the fact that the mouse immunology immune system does not reflect human immune system that well. So studies that work in mice don't work in humans. Many phase 2 trials that were failed to be reproduced in phase 3 trials. And one part of that is that we really don't have reliable or predictive biomarkers that represent a surrogate outcome for determining whether a drug works or not. And this leads to the idea that unlike oncology studies, in which the genetics or gene expression are used to stratify patients for trials, ARDS has not utilized an approach where we subphenotype patients and then use that information to stratify to test a novel therapy. Well, the gap in discovery between discovery and utility of biomarkers was highlighted in a paper we published a couple of years ago. But importantly, to give credence to the idea that NAMT might in fact be part of a strategy in stratifying patients, we measure ENAMP levels in patients with ARDS, trauma, and in fact COVID-19 positive subjects. And you can see, compared to healthy controls, plasma levels of ENAMP in the blood were significantly increased in those patients. We also measured whether there was a difference in severity drove the level of expression. You can see non-hospitalized COVID patients have much lower levels of ENAMP in their blood than hospitalized COVID-19 patients. And we have used the plasma levels of ENAMP to derive a predictive ARDS severity biomarker, in which case ENAMP levels in combination with MIF and IL-8 and IL-6 are able to successfully identify high-risk patients that are at high risk of dying of ARDS. And these levels link to Apache 3 scores, so that ENAMP levels and Apache 3 scores are in fact significantly associated. The other strategy to use for subphenotyping patients is through genetics. And so we sequenced the NAMT gene, and we identified these 5 polymorphisms in the NAMT gene that are associated with individuals' increased risk of developing ARDS and ARDS severity and mortality. And we think by using these 5 SNPs in a genotyping assay that we'll be able to identify patients that are at high risk for ARDS mortality, and as a mechanism of identifying patients most likely to respond to an antibody therapy. And so this schematic basically summarizes that, so that patients coming into the emergency room with impending respiratory failure, be it septic shock, trauma, pneumonia, or COVID-19 infection, can get screened with a rapid point of care test which measures NAMT in the blood, and in doing so, or a genetic test that measures the polymorphisms that make that patient a high-risk patient. And then we can select those patients for a clinical trial where we deliver our antibody prior to intubation, and therefore we've selected high-risk ARDS subjects most likely to respond, and in doing so, and by giving the antibody therapy before placement on the vent, we can abrogate the likelihood of developing ventilator-induced lung injury, can attenuate the cytokine storm that's associated with ARDS, and therefore decrease multi-organ failure and improve survival. Now, just to close here very quickly, I think one aspect of the post-ARDS survivor that's been very, very well illuminated subsequently is the long-term effects and the development of fibrosis in many of these COVID-19 ARDS survivors. That is not the fact that toll-like receptors are involved in pulmonary fibrosis is not a novel concept. A number of papers have shown that. But we believe that the ligation by ENAMT of the toll-like receptor 4s may in fact also be contributing to the likelihood of long-term fibrosis in these ARDS survivors. And we've evaluated toll-like receptor, NAMT, toll-like receptor 4 signaling in a number of disorders, and we have identified the fact that this ENAMT and toll-like receptor 4 is involved in multiple examples of organ fibrosis, which include radiated lungs, includes idiopathic pulmonary fibrosis, and in hepatic fibrosis as well. And this is a paper we just published looking at ENAMT TLR4 signaling in a mouse model of radiation-induced lung injury. And again, I'll bring your attention to Drs. Martin and Liu, who were both important collaborators on that project. And this is just to demonstrate that in that fibrosis model in mice that we have very strong effects of our monoclonal antibody in reducing injury, inflammatory cells, trichrome staining, which is a reflection of fibrosis. Smooth muscle actin is also a reflection of lung fibrosis, which is decreased by the antibody. And when we look at gene expression in these mouse lungs of radiated-induced fibrotic lungs, our antibody primarily affects genes that are involved in lung fibrosis. So in summary, what I've tried to show you is that we've used a very systemic biology approach, looking at genes and proteins and polymorphisms in those genes, post-translational modifications in those proteins, as well as engineered mice and animal models of preclinical and clinical disease, coupled to polymorphisms in human ARDS. And in doing so, we hope to be able to bring a precision ICU approach to the reduction of ARDS mortality. So I'm going to stop right there and just want to thank again Dr. Massoud and Dr. Martin for the invitation to be here and share this information with you. Thank you.
Thank you very much, Dr. Garcia, for a very excellent presentation and information. I have quite a few questions that I'm going to ask from the audience. The first question is ARDS encountered in COVID different from pre-COVID ARDS?
I think the answer to that is unequivocally yes. I think there is classic ARDS observed in COVID-19 patients, in which those patients with the ARDS have lots of lung water, stiff lungs, and behave very much like the pre-COVID ARDS patient. But there's a subset of patients that develop ARDS with lung water that have tremendous hypoxemia, that don't have that stiff lung early on like the ARDS patients. The other thing I would say is a little bit different in the COVID-19 ARDS patient is the propensity for the vascular coagulation complications. ARDS has always had microthrombi as part of the ARDS pathology, but the levels of thrombi and microangiopathic leading to the idea that maybe there's an endothelial vascular endotype in ARDS is something that's kind of unique about the COVID-19 ARDS.
And the next question is, were any gene expressions done between supine and prone position, either human or animal models?
Yeah, so we have a hard time getting volunteers for the human studies that would allow us to sample their lung tissues there, but we looked at, we did supine and prone in the dog models that I shared with you, and there's very, very big differences between the position of the animal and the gene expression, just like you would imagine with the amount of lung water and edema in those particular sites.
Okay. And the next question is, at what stage of ARDS would you expect to use these antibodies?
Well, that's a very, very good question. We believe that this antibody would be useful at almost at any part of the course of a patient with ARDS. Why do I say that? Is that once you're on a ventilator for 2, 3, 4, 5, or 6 days, every day you're on the ventilator is inducing NAMT secretion, so it's exacerbating the inflammatory response. So giving the antibody to neutralize that would be helpful. But for purposes of showing a strong effect of the antibody, we want to give this antibody at the time the patient's intubated. That golden hour. I think it's doable nowadays. Maybe 20 years ago it wasn't doable, but the way ICUs and ERs work together now, I think it is quite doable. And in doing so, I think if we can take the ventilator-induced inflammatory cascade out of the equation, the level of inflammation will be lower, the amount of lung water will be less, and that these patients won't be on the vent as long.
So considering that, now we discuss about the hyperinflammatory and hypoinflammatory ARDS. So at the time of intubation, if we neutralize those antibodies, does that mean it does not make any difference whether the ARDS hyper or hypoinflammatory?
That's a very, very good question. We have no data on this, but my bias here is that by treating the ARDS patient with this antibody, you could shift them from being a hyperinflammatory patient to being a low inflammatory patient. We think that this molecule is such a master regulator of this inflammatory response by activating Toll-like receptor 4. We think that's a big driver in those patients that are hyperinflammatory. In fact, those are the patients that when we want to choose and stratify for a clinical trial, one of the reasons is that we would love to do that trial primarily in hyperinflammatory patients. Moderate to severe ARDS, hyperinflammatory phenotype.
And the next question is, do you anticipate this antibody to have positive impact on inflammation-mediated injury on organs other than lungs? And how will you measure the impact?
Yeah, that's another great question. And that's really what led us to develop that porcine model. And in that porcine model, I think the data I shared with you around renal injury is very, very impactful to me. That showed that within a 12-hour period of a septic shock, ventilator-induced lung injury exposed animal, you see very, very strong evidence of renal injury. And what I shared with you during the course of the talk is that our antibody, given 2 or 6 hours after onset, reduced the level of injury as reflected by lipocalin biomarker for kidney injury, and the histological changes were very dramatic to us. So we think in humans, looking at organ dysfunction, the SOFI scores, in biomarkers of renal injury, as well as liver and cardiac indices, we think that they would show an efficacy of the antibody in preservation of other organ injury.
And I also have a question. For the NAMT pathway, it does prevent the apoptosis of neutrophils. So if we neutralize these NAMT, does that mean that we have an increased activity of neutrophils? And what about the NET formation, which contributes the microthrombosis and the endothelial injury?
Well, so what you don't want in ARDS is a long-lived neutrophil, right? I mean, if you have a massive infection, of course, the amount of inflammatory cytokines and neutrophil influx will greatly overwhelm any biologic therapy. So we think that this antibody will in fact reduce the longevity of these neutrophils, and therefore that's one mechanism by which it reduces the overall level of inflammation. But again, my bias as a vascular biologist is that neutrophils are a major cause of permeability of the microvasculature. Anything that reduces permeability is going to be very beneficial to the patient. So whether it's a reduction of neutrophils or whether it's a reduction of signaling of toll-like receptor 4 on the endothelial cells, we think that that is a very important part of how this antibody works.
And you mentioned in your presentation that the protocol that you are working with is sending the biomarker levels just before, at the diagnosis of ARDS or just before intubation or placing. So does that mean, since mechanical stress can induce inflammation, should we be rechecking those biomarkers after the patient is intubated? Or the first level would be?
So here's what I can tell you, because we've done these studies in ARDS patients as part of the ARDS network and whatnot. If you do sequential daily biomarker studies, NAMP levels go up as the patient goes on the vent. So I'm pretty sure that measuring NAMP levels will just tell you that the patient's on a vent and the vent's driving NAMP expression, and which contributes to inflammation. We believe that if we were to treat people with the antibody before they go on the vent, I think we can abrogate that rise in NAMPT. And I think that that's a very, very important part of how to keep the, inflammation. Controlled inflammation is a good thing. Uncontrolled, unchecked inflammation is a very harmful issue that leads to multi-organ failure. So delivering the antibody at the time of intubation, we think will keep the inflammatory response under check.
Thank you very much. Do you have a question, Dr. Hart?
And Dr. Garcia, could you speak beyond the pulmonary focus? There are other organ systems and disease processes that have inflammatory pathways as a common pathway in those diseases. Brain, for example, in terms of ischemic injury and reperfusion injury and stroke, same for myocardial infarction, reperfusion injury. Chronic inflammatory processes such as those affecting the liver or the gut in inflammatory bowel disease. How far-reaching do you think the pathways that you're studying may be seen in those other disease processes, and by extension the impact potential for the therapeutics that you're developing?
Right, well, appreciate that question, Diego. I have to say, it was obviously serendipitously that we identified NAMPT as a target. It was through that, genome-wide search, and we found a gene and a protein that no one had ever looked at in the inflammatory response. As it turns out, its ability to activate toll-like receptor 4 it's probably the most potent activator of toll-like receptor 4, because it doesn't need these cofactors I talked about earlier. Once you have toll-like receptor 4 as being a part of the signaling pathway that eNAMPT and toll-like receptor 4 are involved in, that just opens up all of innate immunity. So the truth of the matter is, we have done a lot of preclinical studies ranging from inflammatory bowel disease, prostate cancer, because inflammation is ubiquitous. Inflammation drives pathobiologies across acute and chronic spectrums, as you well know, including neoplasia. So wherever you have inflammation, you are contributing to the level of severity of a disease. And it turns out toll-like receptor 4 is a main player. I shared with you a little bit about organ fibrosis. We've looked at kidney, hepatic, lung, and cardiac fibrosis, and this toll-like receptor 4 eNAMPT signaling is important there. We have published a couple of papers on prostate cancer where the hypoxic environment in the prostate drives NAMPT expression, and NAMPT expression drives the aggressiveness of that prostate cancer and the number of metastases. So the ramifications of this signaling pathway, I think we're trying to get our arms around, but we think it's very important, and we think that like a TNF neutralizing therapeutic approach that we see with Humira and other strategies there, we think targeting extracellular NAMP will be an important part of, if not solely as a biologic, but as a part of a cocktail. As we know, ARDS is very complicated. I don't believe that you're going to dramatically move the needle on ARDS with a single therapeutic agent. You're going to need a cocktail just like they have in cancer. But we think this neutralizing eNAMP with this antibody could be a very important part of that cocktail, and I think it could be in other chronic diseases such as you've suggested.
Okay, thank you very much, Dr. Garcia and Dr. Martin. Our next grand round will be on July 30th on acute RV failure and its management, presented by Dr. Bhimraj. Thank you.